Salim sir.
ഓക്കെ സാർ, കേൾക്കുന്നുണ്ട് സാർ.
കേൾക്കുന്നു.
ഓഡിയബിൾ
ആ
സാർ. സ്റ്റാർട്ട് ചെയ്യട്ടെ?
ചെയ്യാം.
Good evening to one and all. Okay, sir, audible?
Yes,
Sir, audible?
Yes, audible.
It gives me immense pleasure and privilege to extend a warm and cordial welcome to everyone to webinar series number 57, jointly organized by IQC Marbasalis College of Education, Sultan Batheri, in collaboration with Association of International Researchers of Indian Origin, ARIO, and the Council for Teacher Education Foundation, Kerala Chapter, CTF. Today's webinar, Understanding Cancer, a Bioinformatics Perspective, focuses on a topic of immense scientific and societal importance. The integration of bioinformatics with cancer research has opened new frontiers in disease diagnosis, treatment, and personalized medicine. We hope today deliberations will enhance our understanding of this rapidly evolving field. It is my proud privilege to extend a heartfelt welcome to our esteemed resource person, Mr. Abunashar Ayub, Master Scholar in the Department of Computer Science at Jamia Millia, Islamia Central University, New Delhi, and Fellow at CLMLMS. Sir, we are truly grateful to you for accepting our invitation despite your busy academic schedule. Your expertise and research experience in bioinformatics will undoubtedly make today's session highly informative and intellectually enriching for all of us. We warmly welcome you to this webinar. Okay, it is my honor to extend a respectful welcome to our beloved principal, Dr. Sunit Kumar Jian.
Thank you, sir.
It also take this opportunity to extend a warm welcome to our distinguished dignitaries. We are honored by the gracious presence of Dr. M.S. Geeta, Chairman of the Council for Teacher Education Foundation, CTO, an eminent teacher educator and academic leader who has made significant contributions to teacher education, educational research, and professional development. Madam, we are delighted to have you with us today. My respectful welcome also goes to Professor Dr. Rajeshwari K, President of ARIO Kerala Chapter, a distinguished academician and researcher who has consistently prompted research culture, innovation, and academic collaboration through her dedicated leadership. Madam, we are privileged by your presence. I also extend a warm welcome to all the members of IQC, ARIO, CTF, Kerala Chapter, faculty members, research scholars, students, and all the participants who have joined us through YouTube. Your enthusiastic participation makes this academic gathering meaningful and successful. With these words, I once again extend a heartfelt welcome to everyone present. I now respectfully invite our esteemed principal, Dr. Sunit Kumar Jian, to kindly preside over the proceedings of today's webinar. Thank you, sir. Sir. Hello, sir. Hello, sir.
സാലിം സാറേ ഒരു മിനിറ്റ് നിന്നോ, സാറിനെ ഒന്ന് വിളിക്കാം.
Okay.
Hello, sir.
Okay, sir. Just give me a minute. So my screen is visible, sir?
Yes, sir, visible.
Sorry, sir.
Thank you. Thank you. Thank you, sir.
Okay.
So once again, good evening, everyone. So today my topic is understanding cancer, a bioinformatics perspective, demystifying cancer through the language of genes, DNA, and data. So in this, I'm going to discuss about the basic understanding of cancer and a bioinformatics and how the bioinformatics helps in cancer treatment. So moving on to the next slides. So before discussing what is cancer, let me tell you something about the cells. So we know that the cell is a structural and functional unit of life, and we all know that human body is made up of trillions of cells. So there are all the trillions of cells, all are basically on normal cells. So these cells basically follow instructions given by the body, or we can say the instruction manuals, that is DNA. So here I'm giving the analogy that DNA is equal to instruction manual. So the normal cells are commanded or given instruction by the instruction manual to divide, that is to replicate, and to stop replication. And then after at certain point they die when they gets old or gets injured. So this is called the this is mainly the function of the normal cells. And what is the cancer cells? The cancer cells are basically just opposite to the normal cells. It ignores all the signals that is given by the instruction manual, that is DNA. It divide non-stops and invade other tissues, other organs. So I can just give an analogy of basic understanding that takes an example of a father and a kid. So father is an instruction manual and it's commanding or giving instruction to its kid. So if kids is obeying or just following the instruction given by its father, so this is considered as a normal cells. But if kids is not following the instruction given by its father, then it is considered as a cancer cells. So, what is a tumor? So, talking about tumor is when millions of cancer cells clump together and then it forms a lump, then it is called a tumor. So, here it is, I have written that cancer is not a single disease, it is a collection of over 100 disease. So, cancer is not just a single called as every cancer we call a cancer. It is not a single disease. There are different types of cancers. Like we can say there are blood cancers, breast cancer, ovarian cancer, pancreatic cancer, and many more. So moving on to the next slides. So this is the 5 steps that follow normal cells and how it works. So the first is the growth signals that the instruction manual gives command to the normal cells to divide, that is replicate. So it replicates. Second is the making a DNA copy. The cells is instructed to make an exact copy of its DNA. And then further third step is to make identical cells. One cells becomes two identical cells. And then fourth is to gives a command, the body gives a command to the normal cells to stop replicating. And final stage is that the program death. When the cells become old or when it gets damaged by some injury, so it do self-destruction, that is apoptosis. So this is the 5 steps that the normal cells follow. So our body is having a more than 37 trillion cells, and they all follow these strict rules. So in the below bottom, there are some facts about these normal cells that are 37 trillion cells is much more than a stars in the Milky Way galaxies. Cell division that our body makes around 3.8 million new cells every seconds. Mostly they are blood cells. And there are checkpoints also when the cells are divided. So when they replicate and make identical copies, so they check the quality of the cells that they are good or not. So checking the errors and the filter, then they allow the spread of the cells. So apoptosis, as I already said, that this is a cell suicide or self-destruction of the cells when the cells are damaged voluntarily or when it gets old. So how cancer starts with a DNA typo. So here are some analogy that I taken. So cancer starts with a DNA typo. So DNA, as I introduced, is an instruction manual. So having a 3 billion letters in it, it is composed of these 4 letters. DNA is equal to ATGC. So these 4 letters make a DNA chains and it contains around 3 billion letters of these 4, combining these 4 letters. So when this letters is changed by this X, which is a mutation, which is called as typo. Here I given analogy as a typo. So when mutation occurs, this X is replaced the ATGC with this X get replaced by this ATGC and cause the cancer, that is mutations. So genes hold the instructions, which gives the more instruction about the DNA, that how the cells behave. So this way, one letter changing in our DNA chain can lead to a cancer. So there are two terms, oncogene turns on and tumor suppressor turns off. So when the cancer cells are, when the cells are converted into a cancer cells, so these two gene cells are responsible for cell growth and cell stoppage. So this gene stuck in on mode, which leads to cells to grow continuously without stopping. And tumor suppressor turns off means its mutation silence the tumor suppressor, which leads to uh, not stopping the cells to stop growing. Next is how cancer begins. So this is this long and slow process, but here are the 6 steps through which a cancer from the initial stage reach to its final stage. So initially it starts with the single mutations that I showed in the previous slide, that there were the chain of DNA in which all these 4 letters were arranged in a sequence. So first it starts with a single mutation. Initially, one letter is replaced with X, that is a typo. Then further it extend to a more mutation, more letters are replaced with X. Then further cell starts behaving like a cancer cells, and it starts avoiding the instruction given by the instruction manual, that is DNA, and start ignoring the rules. Now it leads to a pre-cancer, that it start behaving like abnormal cells. Then further it leads to a cancer cell where it fully ignores all body rules, that whatever the instruction given by the instruction manual, it all ignores the rules, and it all focuses on independent growth, right? Then further it reach to its finest destination, where it forms tumors, right? So this process is basically takes 10 to 20 years to develop from mutation, single mutation, to symptoms. So this is why the early screening is done, early detection is done to stop the cancer in the early stage and not in the later stage. So these are the causes and risk factors. Generally, we people as knowing that the cancers are basically due to genetic variations and due to the environmental factors. But there are many other reasons of causing cancers. So genetic is basically, we are aware of that we form the family history of cancer. If somebody is having a cancer in their family, so it will be inherited to the next generation. And there are some environmental factors through which, if somebody is tobacco smokes, or having taking the UV radiation from the sunlight, or in a environment where pollution is so much, so this can also lead to a cancer. There are some other factors like infections, HPV, which leads to a cervical cancers, hepatitis B and HBC, which leads to a liver cancers, and H. pylori, which leads to a stomach cancer. This is through viruses. And lifestyle is also a factor through which individual can leads to a cancer. So these are the lifestyles. If the individual is consuming alcohol, having a poor diet and obesity, or the physical inactivity. Age is also a factor. Most cancer appears after 15 in the patients we used to see. And here are the external radiation. These are the instrumental radiation. Basically, this radiation comes from the machines which release radiations such as X-rays, CT scans, radon gas, and the nuclear exposure. So this is very important, that hallmarks of cancers. Basically, this is identified by the two scientists, Hanahan and Weinberg. They identified these 8 superpowers, hallmarks of cancer, which cells develop during the growing phase. So when the normal cells convert into the cancer cells, so they have their this 8 hallmarks, the superpowers they developed. That is, they developed a self-sufficient growth. They have their own growth signals. They do not follow any instruction manual that is given by the body or the DNA. They grow on its own signals, on its own instructions. Then it ignores stop signals that was given by the body or by the instruction manual, that is DNA. So it only do cell division on its own basis, not with any instruction given by the instruction manual. Then it escapes cell's death, that we see in the normal cells, that they at the 5th stage they went to the programmed cells, that is apoptosis. When they gets old, or when they gets damaged, the cells do self-destruction, that is apoptosis. But here, when talking about the cancer cells, it disables self-destructs, becomes immortal, where this cancer cells escapes this program. Then 4th is the limitless replications. In normal cells, we see that there is a limit of replication. When instruction is given to stop the replication, there the normal cells stop the replication. But here there is a limitless replication, and the replications goes on forever in a cancer cells. And in a normal cells, it approximately goes for 15 times. Then they have their own builds blood supply. They grows new blood vessels to feed itself, that is angiogenesis. And they also attacks different tissues. They enter the different organs and attacks and spread over the other organs also. So this is metastasis, which is about the spreading of the cancer from the origin to the other organs. So, and last is the tricks the immune system. The immune system is a defense mechanism in a body, so which attacks the any foreign particles and the any unwanted viruses. So it tricks it and make the cells safe, hides from immune cells or switches them off. So these are the 8 hallmarks of cancers. And next is the types of cancers that there are so many, that is 100 plus cancers are there. So here I only cover the major cancers, and these are the scientific name of the cancers, which you can also get idea from the figure that this is a carcinoma, which is a breast or the lung cancer, comes under the breast and lung cancers. This is a sarcoma, which starts in bones, soft tissues. Then is leukemia, which is a blood cancer, cancer of blood cell. It starts in a bone marrow. Next is the lymphoma, which is a cancer of lymph, that is immune systems. Next is myeloma, that is cancer of plasma cells. And the last is the brain, the CNS, that starts in brain or spinal cords. So one more thing, that is the cancer names are given on the basis of the origin from where they started growing. Like for example, the cancers initially or started growing, the origin of the cancer is the lung. From there it started, and then further it spread to some other organs like pancreas and other places. So the name of the cancer will be called from the origin, not from the places where it spreads. So the name of the or identified by the cancer as the lung cancer, not as the other cancer, the pancreas. Though it reached to the pancreas, though it reached to other organs also, spread to the other organ also, but the name of the cancer will be from the origin only. Next is DNA. The analogy of the DNA I gave is the instruction manual, and gene is the instructions, and mutation is the typo. So our cell, as I discussed, is the basic unit of a life and a structured and functional unit of life. So and nucleus is the center of the cells which control, and these are the information like chromosome 46 packages of DNA in cells. DNA is having 3 billion letter codes with all those 4 letters A, T, C, J, G, and gene is like 20,000 genes are total. So these are types of mutation that cause cancers. The proto-oncogene that I earlier discussed, the oncogene, which is for the a gene which is responsible for the switch on of the cell growth. It switch on the cell growth instruction, and the tumor suppressor loss is the switch off the cell's growth. So this is completely, they are stuck on mode, and this also this oncogene is stuck on mode. It means the cells are continuously dividing, and this tumor suppressor loss is in which the gene gets broken, where there is no instruction given to stop the dividing, that the replication of the cells. So here the cancer cells continuously grow. Now the third is the DNA repair gene failure. This the gene that fixes error in DNA, that the mutation, it stops working. So why every cancer is different? So every cancer is considered as different, though it is called the same cancer. Like here I have taken an example that 2 patients both are diagnosed with the same cancer, that is breast cancer, but may have completely different mutation in a DNA, different genes affected, and different immune responses depending on the patients, and need different treatment. Earlier it was like if the patient is going with the any specific type of breast cancer, the treatment was the similar for the same patient, for the both patients. But when here the patients, if it is considered as the both with the same cancers, but their treatment is the different, because the inside the DNA there is a unique changes in the genes and the unique changes in the codes. So due to different mutations inside the DNA, tumor heterogeneity, and due to immune system varies. This immune system plays very important role, because it depends on the patient to patient. Some patients have a different immune cells to fight. Some patients have weak immune cells that they don't fight. So according to that, the different treatments are given to the same type of cancer patients. So environment and epigenetics is also responsible, like with genes are switched on, change based on lifestyle and environment basis. So what is bioinformatics and how the bioinformatics help in cancer disease? So bioinformatics is a multidisciplinary subject in which it combines all the subjects like biology, computer science, mathematics, and statistics. So bioinformatics is a superpower tools which gives scientists to analyze and interpret data, biological data, to find the genetic code hidden in cancer cells. So with the help of bioinformatics, these are the things that we can perform and find the in a cancer cells, like find mutations. So we scan with the help of a program, with the help of computer, we scan billions of DNA letters to spot cancer causing genes. Where this in a billion of letters, where this mutation occurs, like the typo replace the DNA letters, the genetic alphabets. Now it spot patterns also. The normal the is compare with the 1,000 patient to find cancer having in common. It map active genes and it predicts with the help of AI, with the help of machine learning, it predicts which treatment will work best for the specific patients. Then with the help of bioinformatics, we are able to design drugs, targeting medicines and hit cancer. Then it is like early detection. It helps in early detection. Find cancer DNA floating in a blood sample before symptoms appear. So early detection, I will discuss further. This is also a process by which we can do early detection by collecting tumor biopsies or the blood samples. So this is like the Google Maps of biology. Just as we know that Google Maps make sense of millions of roads. In Google Maps, we see that there are millions of roads. So bioinformatics also makes like that, only the billions of biological data are here. So DNA, as I said, is equal to instruction manual, having all the instruction, all the information of an individual in that. Gene is here I'm taking another analogy, that gene is equal to one recipe inside the that manual. Mutation is the typo in the recipe. Cell is equal to a worker flowing those recipes, and bioinformatics is the cell spell checker. So what bioinformatics, with the help of bioinformatics, we check the spell or in the DNA where this X letter mutated gene, sorry, mutation occurs in a DNA, which where it is where it replaces the genetic alphabet. So this is the workflow of the bioinformatics. So here 5 steps are followed from blood sample to insight. So initially, first we collect blood sample from tissue or tumor biopsies from the patients. Then we sequence the DNA. We read all the genetic letters. Then further we store the data and then analyze with the help of the computers with the bioinformatic tools, and we find patterns and typos. Then further analyzing all the data and finding the typos, the doctors get the insights, and further it gives a action plan. Now this how the cancer data generation is done. So there are huge amount of data generated from the patients, and it is like one patient around generates one TB of cancer data, that is and it's like a 500,000 books. So this much of data is generated from the single patient. So this data comes from the different aspects, like patient biopsy, in which the tissue sample is taken from the cancer patient, and then it can be collected from the blood test that catches cancer DNA floating freely in the bloodstream. From medical imaging also, the several medical imaging is done. CT scan, MRI is done of the cancer patients, and photo of the tumor is taken. So from there the data is generated and it is stored. So lab sequences also, where machines read every single genetic letter in the DNA. So genomics in cancer. So genomics is basically a study of the DNA. So in this, reading the instruction manual for mistakes. So here we read the instruction manual, that is DNA, for mistake where this typo error has occurred. So analogy here is, imagine a 3 billion letter book. We are having that in a DNA, and genomics finds every single typo. Even one wrong letter can cause cancer. So even if there is a single alteration that we discussed in the single mutation, that can also lead to a cancer. So here is some R information that I already discussed, that human genome is equal to 3 billion letters of DNA. Mutation is equal to wrong letter, and genomics scans for all typos in a cancer patient's DNA, and it's find exactly which mutate gene is driving the tumor. So transcriptomics is a study of which basically RNA. So listening to which recipes the cell is cooking right now. So our transcriptomics gives a idea of which genes is right now expressing the data. So here the DNA is the full recipe book, as we discussed. So transcriptomics sees where recipes are active right now, or like checking which burners are on. Cancer cells do what they switch on the wrong burners, and we get these much information from transcriptomics, that which genes are turned up too loud, which genes are switched off, or how cancer rewires the cancer cells behavior. And with the help of transcriptomics, we find the drug targets which is hidden in gene activity. Next is how bioinformatics fight cancers. So here are the 6 steps from the step by step. So first we sequence the tumor's DNA and we read every letter. Like we analyze the letter and use to find the typo error in that. Then we compare to the healthy DNA that we have, and we find the mutations in that. Then we ask for the typos are actually driving the cancer, which typos are actually driving the cancer, like which mutation is actually driving the cancer. So we search the global database which has the typo or there the mutation having databases been seen before. In that we search for it. Then further we further match a drug to the typo. We match a drug to the typo, and like a key to a lock, we match it. Then we monitored the tumor function, that the tumor after the drug action, is it shrinking the tumor, or it is like a tumor is outsmarting the drug, that the drug is not affecting showing the effective result. So here, after all this process, the scientists or the doctor came to precision medicines after analyzing the individual patient's cancer cells, and then the right drug is designed for the right patient at the right time. See, old approach was this same chemotherapy was given to every patient, like uh, every patient, if it is suffering from any cancer, different types of cancer, all the patient have to go through the same treatment, like the chemotherapy. Then further, like it is like giving everyone to the same shoes, which is not a good way of approaching a disease. Then old approach was like when the patient goes through the chemotherapy, it kills cancer and healthy cells also. And there were many side effects of this old approach. What were they in the precision medicines? There is a tailored treatment to every individual according to their DNA, according to their immune response, and according to their environment. This is like a custom shoes built for your food. So every individuals are getting a specific treatment according to their mutation, according to their DNA, according to their immune response. And fewer side effects are seen in this precision medicine, and better outcomes are seen. Artificial intelligence in cancer. So artificial intelligence is basically help scientists to analyze or interpret the cancer cells much faster. It is like a radiologist who has having a 10 million x-rays and never get tired or distracted. It is having so much of information about the cancers that it leads to a very fast interpretation and analyzation of the cancer. So these are the 4 steps which follows, like it scans the data, detect cancer in medical images faster and more accurately than humans. Then it predicts response, forecast if patient tumor will respond to a specific drug or not. According to that, the drugs are designed. Then it finds hidden patterns, like in spots connection across millions of patients and record invisibly. Then early detections are done, like diagnosis cancer years before symptoms appears on a scan. So with the help of artificial intelligence, AI, we can do early detections. Next is liquid biopsy, that is early detection using the blood samples. So how we can detect cancer from a blood test? So when a cancer cells die, they release a tiny fragments of DNA into the bloodstreams. So by collecting a few amounts of blood from there, we can just analyze that fragments for DNA, that is ctDNA, which is a circulating tumor DNA. We can isolate it, and then further we sequence it using next generation sequence and read those fragments. And then further we analyze the data using bioinformatics, and we find that typo in that mutations. Then further we reach to our destination, that we get our results, that how the personalized report are made on the basis of the patient specific cancer. So why this liquid biopsy is important? Because this is very non-invasive, when surgery is not needed, and it is very early detection, catch cancer very earlier stage. It tracks treatment, like if some drugs are given for the treatment, so it is working or not, it tracks the records of it, and it is affordable to scale to millions of people. This is the real world case study in a breast cancer and HER2, where patients were diagnosed with aggressive breast cancers, and its DNA cancer cells tumor was sequenced. Bioinformatics finds the HER2 gene overactive and like a broken grow now switch. So AI database matches it and drugs herceptin and targets HER2 directly. This way, then liquid biopsy monitors blood monthly basis that tumor DNA is shrinking, or it's not affecting with it, not suitable for the drugs or not. So further, after reaching to the end, the results shown was the 30 to 50% better survival compared to old style chemo alone. Next is the challenges and the future that the scientists are facing right now in the cancer research, that this patient DNA is deeply personal and privacy is critical, and hospitals don't easily share the data with each other. And if we analyze the data only with a few patients, so the AI can be biased, and it trained on the limited patient groups. So this is also the challenge, that expensive sequences was not available everywhere. And what's the coming next in the future? So cancer detection is going to be very easy, and it is very affordable by blood test at home. And AI designs new cancer drugs entirely from scratch. And custom made immune cells fight your specific tumors will made and cancer treated before it even forms a visible tumor. This is the summarized journey that I discussed in this, like bioinformatics, how the Google Maps of biology workflow, starting from the sample to reaching to the insights, data generation, how the data cancer data is generated from individual patient with the multiple process. Then genomics, finding typo in the DNA, transcriptomics, which genes are on or which genes are off. Then cancer superpowers, that 8 hallmarks we discussed, and the bioinformatics fights, how the bioinformatics fight the cancer by matching the drug to typo. Then precision medicines, that the custom shoes, that the custom treatment is done for the specific patient, though the cancer is the same, but depending on the immune response of the patient's environment factor and the DNA, precision medicine is done. AI, which helps the do the cancer research and the early detection super fast. Then liquid biopsy, which is a cancer from a blood drop, detecting the cancer from a blood drop. These are the key takeaways. That is, cancer is equal to cells that break the rules. So cancer is a cells which breaks the rules and disobey the information given by the instruction manual. And bioinformatics is the multidisciplinary subject, which combines biology, computers, science, mathematics, and statistics. So every cancer is a different. AI is powerful tool, not magic. It basically speed up the process of finding or detecting the cancer. A blood test may soon replaces biopsies. So taking a tumor or samples, it can be replaced by a blood test. By just taking a few amount of blood from the patient can help in detecting the cancer. So the future is personalized. All the treatment in the future is going to be a personalized, not on the um, uh, cancer types on the basis of the patient. So here it comes to the end, and just I want to end with this: like every cancer patient who shares their data makes the next patient treatment a little better. So whatever the data we are getting from the patients, we are just collecting that data, and from that data we are having getting a much more information and getting a benefit of it and treating a future patient a little better. Thank you.
Sanips.
Thank you, sir. Sir.
Conclude ചെയ്യാം.
Okay. Okay, sir. Good evening to one and all. As we come to the close of this enlightening webinar, it is my honor to express our gratitude to all these who contributed to its success. First and foremost, I offer my sincere thanks to our esteemed resource person, Mr. Abunashar Ayub, Master's Scholar in the Department of Computer Science at Jamia Millia, Islamia Central University, New Delhi, and Fellow at CLMLMS. Sir, your insightful presentation on understanding cancer, a bioinformatic perspective, has deepened our understanding of the role of bioinformatics in cancer research. Your scholarly expertise, clear explanation, and engaging in interaction with the participants have made today's session both informative and inspiring. We are truly grateful for your valuable time and sharing of knowledge. I express my sincere gratitude to our respected Principal Dr. Sunish Kumar Chia. I also extended my heartfelt thanks to our esteemed dignitaries, Dr. M.S. Geeta, Chairperson of the Council for Teacher Education Foundation, CTF, and Professor Dr. Rajeshri K., President of Area Kerala Chapter. Your gracious presence and continued encouragement strengthen our commitment to academic excellence and collaborative research. My sincere appreciation goes to the Area and CTF Kerala Chapter for their collaborative efforts in organizing this webinar. Finally, I extend my gratitude to all the faculty members, research scholars, students, and participants who joined us through YouTube. Your enthusiastic participation and active engagement made this webinar truly meaningful. With these words, I once again thank everyone for your valuable presence, support, and cooperation. I wish you all continued success in your academic and professional endeavors. Thank you and have a pleasant evening.
Thank you, sir. Thank you, everyone, for this opportunity.
Okay, thanks, sir. Okay, thank you.